All right, guys. So you guys are welcome to this lecture. We'll be talking about hemochromatosis iron overload. Okay, so we have an overview of iron overload where we spoke about iron as an element. We spoke about the uses of iron. I would say that iron is a nice guy, but if iron is too much in the body, it has some problems, and that's what we call iron overload. Okay, and in the same vein, if iron is reduced in the body, it has some problems. That's what we call iron deficiency. Okay, but for now we're trying to focus on iron overload and talk about the various types of iron overload. Okay, and that's why we are trying to discuss hemochromatosis. Okay, and that is our focus for this video, hemochromatosis. Okay, so basically hemochromatosis is like accumulation in of iron in organs and all those stuff. Okay, accumulation of iron in organs like the heart, like the liver, like the spleen. Do you understand? So guys, but most at times they use the liver to illustrate hemochromatosis. Okay, so looking at this, hemochromatosis can be primary. That's it has no secondary cause. It is primary. Do you understand? Like it's hereditary. Okay, they just gave birth to you with it. Okay, and this can be more of like gene defect. Okay, gene mutation. Okay, and secondary hemochromatosis can be secondary. Okay, so like sorry, hemochromatosis could be secondary, and that's what we call secondary hemochromatosis. And this is basically accumulation of iron in organs that is from the secondary cause. Do you understand? So adding to this, iron can also still accumulate in endocrine organs. Okay, like if iron accumulates in the thyroid gland, in the parathyroid gland, it can lead to hypothyroidism and hypoparathyroidism. That's basically a reduction in the production of the hormones that all these endocrine organs will produce. Do you understand? So secondary hemochromatosis has means that okay, there's accumulation of iron in organs, but it is another thing that is leading to this accumulation. Do you understand? So it is a secondary cause. Okay, and it could be from repeated blood transfusion, and that's what we call transfusional hemosiderosis. Okay, and it could be from the African siderosis. Okay, so let me shed more light on this. Blood transfusion is like okay, some people are having some blood is not to the required amount in the body, so you have to actually opt in for blood transfusion because it is the easiest. Okay, so if you actually have multiple blood transfusions, it has its side effects. Okay, it can lead to iron overload in the body. Okay, and African siderosis is more of like okay, some Africans have gene defect that will actually lead to too much absorption of iron. Do you understand? So if you have problems from your gene on too much absorption of iron, then you are still taking diets that have a lot of iron because Africans take a lot of meat. Let me confess, if there's anything that the doctor will ever tell me that you should stop taking meat, I don't take alcohol, I don't smoke, I don't womanize, I don't do any of those things, okay? But I eat meat, bro. Do you understand? I can't eat food without meat, for real. Okay, so I'm a typical African, okay? And African siderosis is basically a gene defect and mainly the types of diets that Africans take, okay? It contains a lot of iron and it can lead to accumulation of iron in the body. Okay, so I've already explained this slide. So I've already spoken extensively on the secondary causes of hemochromatosis. So this lecture will focus more on the primary hemochromatosis, okay? Hemochromatosis, okay? So we'll be focusing on the gene defect. Let's talk about this gene defect since you guys already know what the secondary causes are. So primary hemochromatosis is also called the hereditary or genetic hemochromatosis. Okay, so it is a group of diseases in which there is excessive absorption of iron from the gastrointestinal tract, leading to iron overload. Okay, do you understand? So there's a genetic defect, and this genetic defect is making you absorb too much iron. Do you understand? So it will lead to iron overload in parenchymal cells of the liver, the heart, the endocrine organs, and also the spleen. Okay, so examples of these endocrine organs, like I told you guys, the thyroid, the parathyroid, the pancreas. Okay, so what is this gene defect? All right, which gene is actually affected? So this results from a point mutation in the HFE gene. And what is the full meaning of HFE gene? That's what the human homeostatic ion regulator. So do you guys see that the full meaning of this is not really rhyming with the abbreviation? I guess like, okay, why is that HFE is not really rhyming up with human homeostatic ion regulator? H here stands for what? Homeostatic. Fe is the chemical symbol for iron. Do you understand? So homeostatic ion regulator gene, that's what you could call it, or you could call it like human homeostatic ion regulator gene. Okay. So the human homeostatic regulator ion regulator gene is a negative regulator of hepcidin. Okay. And what is the function of hepcidin? Hepcidin is a protein formed by the liver, and this hepcidin will actually bind to the proteins that actually allow the passage of iron as ferroproteins and to destroy ferroproteins. Okay, so it will actually reduce the absorption of iron. Okay, but if this hormone that is meant to stop the absorption of iron has a defect, then that means absorption of iron will be too much. Okay, so these are the basis of these things. You guys don't really need to cram everything. Do you understand? So guys, it was first, this primary hemochromatosis was first described in 1865 as a clinical triad of what? Glycosuria, that's too much glycogen in the body. Liver cirrhosis is basically accumulation of iron in the liver. Okay, then hyperpigmentation of the skin, that's basically like too much color in the skin and all that. Okay, so von Recklinghausen later established that these clinical features were due to iron deposition, and he coined the term hemochromatosis, meaning deposition of iron in organs. Do you understand? These organs, let's go over them again: the liver, the spleen, the heart, the endocrine organs like the what? Thyroid, parathyroid, pancreas. Okay, so guys, that's it. I could see this is a healthy liver. I could see this is an iron overload. Okay, so most of the time they use the liver to actually illustrate iron overload. You see that mostly we don't really have pictures of the heart, the endocrine glands. This is because the position of iron in the liver is the most commonest. Do you understand? So still talking about gene involved is what? Human hemostatic ion regulator gene. And this is situated close to the major histocompatibility locus on the chromosome 6. And the human homeostatic ion regulator gene is involved in hepcidin synthesis, and therefore hereditary hemochromatosis caused by human homeostatic ion regulator mutation is due to what? Low serum levels of hepcidin, because this hepcidin is supposed to regulate the absorption of iron. So if this hepcidin is absent, then there will be too much absorption of iron, leading to what? Too much deposition of iron in organs. You understand? So that's the timeline of the whole shit. So low serum hepcidin levels lead to high levels of ferroportin, and therefore increased iron absorption and increased release of iron from macrophages. Okay, so guys, these are the primary basis of what hemochromatosis iron overload is like. Okay, talking about the primary hemochromatosis and the secondary hemochromatosis. Okay, so that's it, and see you guys in the next one. Bye for now.